Hi, my name is Linda and I work in Dr. Edwin Manuki's lab at the University of California, Irvine. Today I'm going to show you how I use Sigma's Cyber Green Extract-and-Amp Tissue PCR kit to rapidly genotype our transgenic mice. Usually I use this kit to genotype our embryonic mice when I'm doing acutely dissociated cell cultures and I need to rapidly and quickly find out the genotype of our mouse. But for today's purposes, we have adult mice tails in which we can show you how I use this kit. This protocol involves a quick tail digestion and DNA extraction, and then a quick neutralization step. And we use the extracted DNA in a real-time PCR where we can monitor our PCR amplicons. So if you're ready, let's get started. So before we get started, we want to make sure that our heat block is set to 95 degrees Celsius, that our Cyber Green is thawed and is kept on ice until it's needed, and that our other reagents are thawing to room temperature. In my ice bucket, I have my 2 primers that we'll be using to genotype, and they've been specifically designed for our gene of interest and validated for qPCR. I also have my positive control for the gene of interest and my negative control, and a tissue sample that I've already collected from an adult mouse. Now, when you collect your tissue sample, it's very important to be extremely stringent. When we collect embryonic mouse tails, we always rinse our forceps with 70% ethanol prior to and before each tissue collection, and we always rinse the tail samples in PBS. First, you have to make a master mix of the tissue preparation and DNA extraction. For each tail, you're going to want 100 microliters of the extraction solution and 25 microliters of the tissue prep solution. I'm going to use my P200 and filter pipette tips to measure out 100 microliters of the extraction solution and 25 microliters of the tissue prep solution into a new Eppendorf tube. You're going to want to mix your master mix solution by pipetting up and down. And so you're going to want to add 125 microliters of your master mix into your tissue of interest, making sure that if it's an adult mouse tail, that the cut end is completely submerged in the master mix solution. And you can finger vortex it, and you incubate it at room temperature for 10 minutes. During the 10-minute incubation, I'm going to prepare my Cyber Green Master Mix, which involves the Cyber Green Master Mix that they include in the kit, milq or PCR grade water, and our primers. Some PCR grade water, and our forward and reverse primers. And that's it. Now that the 10-minute incubation is up, we're going to have to heat and activate the reaction at 95 degrees for 3 minutes. Our 3-minute inactivation is up, and now we're going to add neutralization solution that's provided in the kit, and we're going to add 100 microliters per reaction. And you're going to mix this by vortexing. After you add the neutralization buffer, your DNA samples can be stored at 4 degrees Celsius until you're ready to use them. We actually want our genotype right away, so we go immediately into the qPCR. So my tails are neutralized and are ready. My Cyber Green PCR Master Mix is ready, and I have my PCR tubes set up on a cold block. All I'm going to do now is add 16 microliters of my master mix to each well, and then add 4 microliters of the neutralized extract, or 4 microliters of the positive or negative control to each well. All I'm doing is adding 16 microliters of the master mix to each of the wells. Then I will add 4 microliters of the neutralized extract to the 60 microliters of the master mix in the well for a total of a 20 microliter reaction volume. And I'll do the same thing for the positive and negative control, adding 4 microliters to 60 microliters of the PCR Master Mix. Once they're all added, you can cap them with an optically clear 8-cap strip. You want to be careful to not touch the optically clear part of the caps that are over your samples. So I'm using a Kim wipe to push down on the caps to make sure that they're sealed. So I've gently vortexed our PCR and DNA mix, and I've spun them down, and now we're ready to put them into the qPCR machine. What we're going to do is first we're going to load our samples into the Opticon PCR machine. And I'm going to make a new plate setup. In which our first well will be our sample. Our second well will be our positive control. And our third well is our negative control, specifying that our reaction volume is 20 microliters in total. Next, I'm going to load a protocol that we've designed for for Sigma's rapid genotyping. This protocol involves a 3-minute denaturing step, a cycle of 15 seconds of denaturing, 15 seconds of primer annealing, and then 20 seconds of primer extension. Now, the primer extension time can actually be dropped down to about 10 seconds, and thus you'll have a total of only 40 seconds for each cycle. We have those 3 steps cycling about 40 times like in a normal PCR, and then at the end of that we take a melting curve. So we have our plate set up and we have our protocol loaded. All we have to do now is run our qPCR. And it's starting at the 3-minute denaturing step. From my experience using the primers that I'm using and using the tail samples and looking for the genes that I'm looking for, I know that my positives will start to come up around 25 cycles, crossing a cycle threshold of significance. Now, when that crosses is highly dependent upon your tissue sample and what you're looking for and how much of the DNA is present initially. So we can already see that our positive control has already come up, and it looks like our sample is also positive. As you see, it's starting to come up a little bit after our positive control. We're still going to have to wait it out because it's only at 27 cycles, but hopefully in a couple more cycles we can be sure that what our sample is positive, and I can then proceed with my experiment. At this point you can see the negative control just starting to come up, and that's because I took an excess of tail. That helps you be sure that anything that comes up before this excess negative tail is going to be positive. So at this point, anything that is coming up before the negative, I would consider positive, and I would proceed with my experiment. After the PCR is finished cycling, the machine performs a melting curve analysis. This will help you determine the composition and purity of your product. With this, you can tell if you chose the positive and negative samples correctly. For instance, the positive sample will have a specific sharp melting temperature peak, which corresponds to a specific PCR product amplified. Any negative samples will have a nonspecific broad melting temperature peak, corresponding to nonspecific DNA that had been amplified during the PCR. So I've just shown you how we use Sigma's Cyber-Green Extractant Amp kit to rapidly genotype adult mouse tails, but this kit can be used with various animal tissues, hair, saliva, and I use it to genotype mouse embryonic tails. We like to use this kit because it's rapid, I can get my genotype back in an hour, and it's very convenient and simple. So thanks for watching and good luck.